Hi, everybody here and online. I'm Chris Johnson. I'm an epidemiologist at Henry Ford Health and the Deputy Director of CURES, which, as most of you know, is the NIEHS-funded Environmental Health Sciences Center here at Wayne. It gives me great pleasure today to introduce my friend and colleague, Dr. Patrick Ryan. Dr. Ryan is a professor of pediatrics and environmental and public health sciences at Cincinnati Children's Hospital Medical Center and also the University of Cincinnati College of Medicine. He obtained a BS in mathematics from Xavier, is that right? University, and an M.S. and Ph.D. In epidemiology from the University of Cincinnati in the Department of Environmental Health. In 2013, he joined the faculty of Cincinnati Children's, and he currently wears multiple hats. He's the Associate Director for Research in the Division of Biostatistics and Epidemiology, the Director of the Graduate Program in Clinical and Translational Research, and the Director of the Translational Workforce Development Core for the Center for Clinical and Translational Science and Training. It's a lot.
Yeah.
Whoa. Dr. Ryan's research is, I think, right on target with the CURES audience. He works to integrate exposure science and epidemiology to study the impact of environmental exposures and social determinants of health, including air pollution, community resources, and green space, and all of this on the outcomes of respiratory and neurodevelopmental health in both children and adults. In addition, his research includes the use of personal air monitors and health sensors in epidemiological studies, and even the impact of natural occurring elongated mineral fibers on pulmonary disease, which is in the Western US, these studies. So I've been fortunate enough to work with Pat on a number of pediatric asthma birth cohort studies. Most recently, we've been colleagues in a group called CREW, probably my favorite group ever. It stands for the Children's Respiratory and Environmental Work Group. And this is a consortium of 12 birth cohorts that are a part of the NIH's large research study called Environmental Influences on Child Health Outcomes, or ECHO. So Wayne State, Henry Ford, MSU, and U of M have all been a part of ECHO. And Pat has been a really key player in this program. So I'm delighted that Dr. Ryan is going to present today on air pollution, community characteristics, and children's health.
Thank you. Thanks for the introduction, Chris. So I really appreciate the opportunity to be here today, the invitation to visit Detroit. As Chris mentioned, I really enjoy epidemiology, environmental epidemiology, because we get to do so many different things. I gave the talk a relatively generic title because I really do want to try to cover a lot of different projects and research that I've been lucky enough to be involved with, primarily in obviously environmental exposures and community characteristics in respiratory and neurodevelopmental outcomes, but also try to sprinkle in there some of the work that we've done in exposure science. So how do we actually, in epidemiology studies where we're trying to look at air pollution exposure, characterize what that looks like for kids? So I'm going to talk a little bit about some of the relatively straightforward, simple approaches that we used to do with GIS, to the more complicated spatial models of exposure, to the more recent work that we're doing with personal monitors and low-cost sensor technology. So that's the goal. A lot of different projects, a lot of different topics that I'm going to cover, and I'm happy to chat more about any of them if we have time. So I just wanted to start by framing all of this. I think there's this I shouldn't say it's new, but this general increase in awareness of the importance of social determinants of health. And certainly, I think that all of the things that I'm going to talk about fall under this umbrella. The idea that where kids are born, or where anyone, but I talk about kids because that's what we do, where kids are born, grow, work, live, play, all influences the health outcomes that they have. And certainly environmental exposures, and as an environmental epidemiologist, we have to think about not just what we're interested in, maybe it's air pollution exposure, but the context in which that occurs, because, no one's exposed just to air pollution, but they're exposed to air pollution at the same time that they might be struggling to find a place to sleep that night, or where are they going to get their next meal. So environmental exposures and the context in which they occur play an important role in health outcomes. And as Chris mentioned, I've been lucky enough, I'm going to talk Today, focusing mostly about air pollution, but also we've worked with green space and naturally occurring asbestos exposures and neighborhood characteristics, things like population density and poverty in the neighborhood, but also things like crime and access to care, food deserts. And then now I'm interested really in how do we change things? So how do bad policies make bad outcomes, and maybe good policies could maybe make some better outcomes. So I'm going to start with the Cincinnati Childhood Allergy and Air Pollution Study. So this is a birth cohort study. It's the cohort that Chris mentioned that's part of the CREW consortium and is part of the ECHO-wide project from NIH. The Cincinnati Childhood Allergy and Air Pollution Study, the original hypothesis or the goal of the study was to look at whether or not traffic-related air pollution exposure, and specifically we're interested in diesel exhaust particles for a couple of reasons. If that increased the risk for kids to develop allergic diseases and asthma, and then now we're also interested in how that impacts neurodevelopmental outcomes in kids throughout their life. And so the setup of the study, this is a long-term birth cohort. The kids were born between 2001 and 2003, so they're now in their early 20s, and we are still seeing them as part of some of our ongoing research. But it's a longitudinal birth cohort study. The original design was that half of the cohort had a birth record address that was either less than 400 meters from a major road. So that's what we thought initially would be our exposed population. And then half of the kids would be born living 1,500 meters or more from that major road. And so we follow them longitudinally. You can see here the design of the study. Am I getting feedback? Do you hear feedback? Okay. So the design of the study, we saw them initially at ages 1, 2, 3, 4, 7, 12, 14 to 17 as part of the ECHO project. And we're now still seeing them in their late teens and early 20s. And we've got a lot of data that's collected on them. I'm going to focus mostly on the air pollution exposure, but we've also measured dust or sampled dust and measured things like allergens and mold exposures in the home. And we're interested in obviously multiple outcomes with questionnaires, spirometry, and a number of mental health outcomes that I'll go into. So we've long-term follow-up on this cohort. So this is one of the very first analyses that we did now about 20 years ago. This is looking at infant wheezing. So it's parental report of their child wheezing in the first year of their life. And what I'm showing here is what we did was categorize them using GIS as to how far they lived from major roads and what we called stop and go traffic in the Cincinnati area. So this is what we call a proximity model, and we're just characterizing them or categorizing them into these three categories of exposure based upon where their house was at the time they were born. So we had a group of kids that were unexposed. So those are the kids that lived near or not near any of these types of traffic. Then we had kids that lived where we considered exposed to stop and go traffic. Those are the kids that lived in the blue here, less than 100 meters from one of those stop and go traffic routes. So in, think state highways with stoplights, basically. And then the moving traffic, those are the interstate highways in Cincinnati. So higher volumes of traffic, but more of a moving traffic pattern when not in rush hour, perhaps. And those are the kids that lived less than 400 meters from there. And a couple of takeaways from the results that we saw in the first year of life. First, you see the association between living in that stop and go category and also the moving category at an increased risk for child wheezing, parental report of their child wheezing in the first year of their life. And secondly, and we'll talk more about this too, the racial disparity that exists even in the first year of life in parent report of their infant wheezing. And you can see that across the board in the three categories. So both of those were significantly associated with infant wheezing in their first year of their life. So that proximity model, relatively simple. It has some pros to it. It's easy to do. It's easy to explain to people. And it's easy to think about ways of intervening on something if you're near or far from a major interstate. But if you think about it, and I don't know about Detroit, but in Cincinnati, there are areas of major interstates that have much less traffic than other areas, even though they're all interstate highways. And so what we decided to do was develop a model called land use regression. So this is a technique that takes advantage of the air sampling that we did in Cincinnati at the same time as the cohort in the early 2000s. And what you do in a land use regression model is you have your stationary air samples outside and you take the air samples just like the EPA does. We did filter-based samples. We measured PM 2.5 and we also measured something called elemental carbon, which is a marker of traffic, specifically diesel exhaust. Again, we were interested in diesel exhaust particles. We measured how much of the PM 2.5 and the elemental carbon was on our filters at these 27 sampling sites. And then we develop a model that's basically a linear regression model. And what we do is we try to predict the sampled concentrations at our 27 sites using geographic and other land use characteristics that surround those sites. So in our model, the final model in this approach had the number of trucks that passed by that site within 400 meters, the length of bus routes within 100 meters, and the elevation of the site. And using those three variables, we get pretty good R-square value for our model. And then you can take that information and predict or estimate the exposure, the concentrations at any location in the city. And so that's what you see in the middle figure is just a smooth surface of the model at all the locations in the city. And if you're not familiar with Cincinnati, you can basically see the outline of our interstates. And so, you know, the higher traffic areas, the higher exposure to diesel exhaust particles and traffic exposure, which makes sense. And then you can put those dots on that last figure on the right. Those are the homes, the birth record addresses, the homes of our kids in the study. And the bigger, the darker the circle, the higher the estimated exposure at their home address. And so we can actually quantitatively estimate what's the ambient exposure or concentration of elemental carbon in this case at the homes of our kids in the study. And then when we go to look at health effects, this is from two different papers. The first set of results on the left side of the screen, that's looking again at infant wheezing in the first year of life. So we went back and we said, okay, now what is our quantitatively estimated exposure to elemental carbon? What's its association with infant wheezing in the first year of life? Again, we see a significant association here. I'm just looking at different concentrations. The median concentration for reference in Cincinnati is about 0.4 micrograms per cubic meter. This is elemental carbon. So it's just a fraction of the PM 2.5. And at that level, there's about a 50% increased risk in wheezing in the first year of life in the kids in our cohort. Of course, the advantage of a longitudinal birth cohort is we can follow them prospectively, and that's what we've done. We can start to look at the development of asthma, and that's what I'm showing in that figure on the right. Those are estimates of air pollution exposure in different years of the child's life, so from ages 0 to 1, and then 1 to 2, 2 to 3, and so on and so forth. A couple of take-homes. It's the early exposure that seems to be the most relevant. So those exposures were associated with increased risk of having asthma by the time they were 7 years old, and also that cumulative exposure. The exposures that occurred later in their life, 4 to 6 and 6 to 7, those are the ones that you see there's no association there between the exposure. So early childhood exposure and cumulative exposure seem to be associated in our cohort with the development of asthma by the time they were 7 years old. And we've looked at a number of other health effects, allergic diseases with this model and elemental carbon.
Is it 1 o'clock? Are you guys at 1 o'clock for the next thing?
I'm not going to
okay.
go into it
Start walking soon.
Too in depth.
1 o'clock for the CEC.
As long as I'm not being.
Oh, you can.
Yeah. Yeah. I'm gonna switch gears and talk a little bit about nervous system effects. So in about the mid to late 2000s, there was an increased interest in how air pollution might actually impact brain development, particularly the small particles, so PM 2.5, and then also what we call ultrafine particles. So these are particles in the nomenclature of particulate matter that would be PM 0.1. So these are particles by definition that are less than 100 nanometers in diameter. So orders of magnitude smaller than PM 2.5. The primary source of these are traffic outside. There's also indoor sources. Diesel exhaust is primarily ultrafine, at least fresh diesel exhaust. So we had a cohort that we could start to think about what are the effects of air pollution exposure. We've already characterized that if we start to look at some of the neurodevelopmental outcomes in our cohort. And the mechanisms have been proposed, and there's been, at this point, you know, over the last 15 years, multiple studies showing the mechanisms behind this, both direct mechanisms by which these small particles can directly impact the brain, whether it be through the olfactory bulb or translocation, but also indirect mechanisms. You know, the body doesn't like to breathe air pollution. It produces an inflammatory response, which is the brain also has that response. And so you get these increases in neuroinflammatory markers as well with air pollution exposure. And of course, it's this butterfly effect of lead's the classic example. If you're exposed early in life while the brain is developing, perhaps that manifests itself much later on, or sometimes later on with different outcomes. And so in our cohort, we wanted to look at that at age 12. And this is just showing now sort of the state of epidemiology here. Everywhere you see a red arrow that's down or up, that's kind of, there's been lots of epidemiology showing associations between air pollution exposure and those outcomes. So things like IQ as an outcome, executive function, decision-making in kids. There's been studies of autism spectrum, and so on and so forth. The circle there is what I'm going to focus on today, because less has been done in that realm. In this frame, they're calling it social emotional. I'll use the term mental health, or we will talk about internalizing disorders. Those are things like anxiety and depression. So those are the outcomes that at this point, less has been done. So where do we stand? So in adults, there has been a number of studies that have shown air pollution is associated with some mental health outcomes, things like increased risk for suicide or attempted suicide, increased risk for emergency department visits for depression and anxiety. But kids is where the action is when it comes to these outcomes, right? So the first onset is in the early teens, early adolescence. For most of these, anxiety and depression, it's a major issue, public health issue, mental health outcomes in kids. And so we wanted to start to look at these internalizing behaviors that are difficult to detect. And I think that's part of the reason why there's been less epi done on these. It's hard to get at anxiety and depression. By definition, they're things that are internal, and parents don't often recognize that. But they have lifelong implications and major health impact. So this is just a snapshot of the protocol that we did at age 12 in the CAPS cohort. The idea here is, I'm not going to go through all of these, but it was about a 5 to 6 hour study visit that we did with these kids, including an MRI on half of them if they had high or low air pollution exposure in their first year of life. So we have functional MRI and structural data on the kids with MRIs. And then we also did both self-assessments with the kids themselves when they were 12 years old, but then also parental report of various outcomes. And then also importantly, we also have the caregivers' depression and IQ as well. So this analysis was focusing on the outcomes of anxiety and depression in the kids at age 12. So again, we assessed this 2 different ways. Parent report using something called the Behavioral Assessment System for Children, the BASC-2. It's a validated assessment. You get both internalizing and externalizing behaviors, and also composite scores. Here, I'm just going to focus on 2 subscales, the depression and the anxiety subscale. It's a normalized assessment. So the mean is 50, standard deviation of 10. And then the child's own self-report using 2 different assessments, the Child Depression Inventory 2 short form, the CDI-2, and the Spence Children's Anxiety Scale, or the SCAS. Again, these are population norm with a mean of 50, standard deviation of 10. And in both scales, the higher the number, the more of that symptom that they're exhibiting. A couple of things to note. So first, I have to remember the parents report their children having slightly more anxiety than the children themselves reporting. So parents tend to think their children have more anxiety symptoms than the kids themselves report, and the opposite for the depression symptoms. And the other thing to take note of here is those correlations that I'm showing there in the middle are the correlations between the parent report and the child report. And so again, by definition, parents might not recognize some of these disorders. And you can see the correlation between parents' report of depression and their child's report is 0.35 and anxiety 0.24. So relatively low correlation between the parents' report and the child report. So that's important to know, because when we did the analyses, when we looked at the parent report of their child's anxiety and depression outcomes, we saw no significant associations with our air pollution exposure and those outcomes. But when we looked at the child's own report of their anxiety and depression, we saw significant associations with depression and with generalized and specific subscales of anxiety. And so you can see here just the general pattern. In each one of these, we're looking at exposure in the first year of life, the year before their assessment, and then their average exposure over the 12 years of their life. And you can see significant associations throughout, mostly with the early exposures, some with the cumulative and the later exposures as well for the depression, and then also the total anxiety and some of the other subscales of anxiety. So a significant association between air pollution exposure at various points in the child's life and their self-report of anxiety and depression symptoms. And so based on that, we have a study now where we're actually taking the CAPS cohort and another cohort in Cincinnati called the home study cohort, and we're harmonizing those cohorts together into something we're calling the Cincinnati Combined Childhood Cohort, or the C4 cohort. And the idea here is to take this. The home study was also established about the same time. It used to be part of a children's center grant that was in Cincinnati. There are 468 pregnant women that were enrolled in this study. They're actively following more than 400 of them. And we have co-investigators around both cohorts. So the psychologist that designed all these assessments and the imaging expert that did the MRI protocol for us is the same on both cohorts. So we can harmonize all these outcomes at all the ages of their life. We can use our air pollution models to estimate their air pollution exposures. And we are now looking at and following up with study visits both cohorts in their late teens and early 20s to look at trajectories of all these different outcomes and changes in the MRIs from early adolescence at age 12 into late adolescence and early adulthood. So stay tuned. We have about 350 of our target 500 study visits done right now. So we'll hopefully be done with that in the next year or so with the study visits for the C4 study. So I'm going to switch gears slightly, still focusing on mental health outcomes. But everything I've talked to you about so far has been using that land use regression model of air pollution exposure. So what that model is doing is looking at average exposure over long periods of time, because it's based upon that air sampling that happened back in the early 2000s, 24-hour samples, and we averaged them basically at every sampling site. So you can think of all those estimates of air pollution exposure that I've talked about so far as average or chronic exposures, which is probably okay when we're thinking about the types of outcomes that we were talking about before, these symptoms of anxiety that develop over the first 12 years of their life. But if we wanted to think about, and we wanted to think about what does it mean if there's a bad air quality day in terms of a short-term exposure and then a more acute outcome. So in the asthma world, we think about this a lot. There's an increase in asthma emergency department visits a day or two after there's a bad air quality day. And here we're doing the analogous type of question. Is there an increased risk for a mental health emergency department visit in the days after a bad air quality day? So the way we did this analysis is a study design called a case crossover study design. So it's a good study design when you're looking at short-term exposures and acute outcomes. And what it does is it allows every, in this case, child participant in the study to serve as their own control. So things like genetics and socioeconomic status that don't change over short periods of time or at all in the case of genetics are not confounders in this analysis. So what it does is what we did was we took all the emergency department visits from 2010 to 2014 or 2015 at Cincinnati Children's Hospital. We geocoded them at the time, meaning that we took their address from the electronic health records. We put them on the Earth's surface, and we take advantage of a friend and colleague of mine, Cole Brokamp, who developed this air pollution model that actually is able to estimate the amount of PM2.5 at every 1 kilometer grid over the entire country from 2000 up to 2020 now. And he has a model. There's other models out there very similar to this, but what it allows us to do is actually look at on a specific day, what was the estimated air pollution level at a 1 kilometer resolution. So we can estimate for a child on a specific date, their exposure to PM2.5. And then we can look at, this is the crossover part of the design, 2 weeks before or 2 weeks later when they're not in the hospital, what was their air pollution level at those time points. And the idea here is that if our hypothesis is that there's higher levels of air pollution exposure, or the air pollution is associated with the emergency department visit, the levels ought to be higher in the day or the days before the date that they were hospitalized. So that's the crossover design of this. And so that's what we did. The figures here are just showing you an example of what PM 2.5 looks like. The top figure is the average over, I think, 10 or 12 years in the Cincinnati area. The bottom figure is one specific date, June 18th, 2010. You can see it's a very different picture. So the air quality changes, air pollution levels change day to day. So we can do this type of analysis using this type of model. And here's the results. Basically what we saw was that significant associations between air pollution exposure, PM 2.5 in this case, and increased risk for things like mood disorders, all-cause mental health outcomes, adjustment disorders, externalizing disorders, and suicidality, mostly in the day to 2 days before the hospitalization itself at Children's Hospital. So a significant association between short-term exposure to air pollution and these acute health effects, in this case, a hospitalization for one of these mental health outcomes. And the other thing I want to highlight from this is now turning to the community in which these kids live, the role of the environment, the neighborhood, in modifying these outcomes. And so here, what I'm just showing you is what we did was at the census tract level, characterize whether or not the child's census tract has what we call high or low deprivation based on indicators of socioeconomic status. And here it's just the median exposure or median deprivation index. So if they were high or low, so a higher means more poverty, less access to care. Low deprivation means less poverty, more access to care. And what the takeaway here is the effects are modified by that community environment. So air pollution sometimes increases the risk and sometimes appears to be decreasing the risk, or vice versa. The community seems to either enhance or mitigate some of the effects of air pollution exposure on our outcomes. And this is not unique to the mental health outcomes that I'm talking about here. We've seen it with asthma, and you see it with other indicators of community status. So the effects of air pollution and other environmental exposures tend to differ based upon the underlying risk factors in a child's community. So switching gears a little bit now, focusing more on community, race, and health outcomes. We know now, back to the asthma world, but this is not unique to asthma, obviously. Black and Hispanic children are at an disproportionately increased risk for asthma-related morbidity. The data I'm showing you there comes from Cincinnati Children's Hospital. Let's see if I can actually not use that pointer apparently, but the line that's up on top is I think it's the rate of asthma ED admissions at Children's Hospital for Black children. And then the line at the bottom is for the white children over a 2-year time period. So that's not unique to Cincinnati. I'm sure you probably would see the same thing in Detroit and everywhere else. And of course, there's also disparities in terms of where these kids live. And it's a little hard to see here, but the census tracts that have colors to them, the more yellow-red is the higher rates of asthma admissions in the Cincinnati area. And so there's disparities both in terms of, thank you. Yeah, so there's disparities in both terms of race and in terms of where these kids live in Cincinnati. And so this brings me to the CREW consortium that Chris mentioned. It stands for the Children's Respiratory and Environmental Work Group. It's a consortium of 12 allergy and asthma birth cohorts at 15 different sites. You can see the cluster of them here in Detroit, multiple cohorts in Detroit. But in total, this brings the study population for the consortium to about 7,000 kids. All of them followed longitudinally for the development of allergic disease and asthma. The advantages of this consortium is the temporal variability. So kids in the CREW cohorts have been born as early as 1980, all the way up until they're recruiting now. And there's a lot of asthma. So there's more than 1,500 cases of incident asthma in the CREW cohorts. And so we can start to think about what does community air pollution exposure, race, what does the interplay look like because we have such a diverse study population in terms of geography and time. And so this is some results from a paper that we published looking at children's race and ethnicity and neighborhood socioeconomic position. And so the question that we wanted to ask here was, does neighborhood level SES indicators, does that explain some of that race ethnicity difference in the childhood asthma? And you can see the Kaplan-Meier curves here are just the raw survival curves based upon race and then indicators of the child's neighborhood in terms of poverty, low income. If you can't see it, on the bottom on the race is Black children. And then census tracts with higher levels of poverty and more low income are more likely to develop asthma early in their life. And so because of the diversity of the crew cohorts in terms of geography, we can look at the interaction between race and ethnicity and where they live. And so in addition to looking at just census tracts, we can look at the interaction between race, ethnicity, and that census tract. So these are the results. So this is the hazard ratios that I'm showing here are the association between a neighborhood indicator of SES and the risk of asthma. And what you can see is there's 4 that turned out to be significantly associated after we adjusted for everything. Median household income and gross rent, so the higher levels are protective, which you might expect. And then the percent of low-income households in a census tract and the population of that census tract were both risk factors for developing asthma. And this is through age 12 in this analysis. So the next step of this was, what's the interaction between race and ethnicity? So if we put race and ethnicity and census tract indicator of SES in the same model, look at the interaction, can we explain away some of those differences? And what it turns out is that there was no significant interaction between race and ethnicity and the risk of asthma. And so this is showing in the top half there is for race. The hazards ratios are for Black children compared to white children across categories of their census tract SES. And the take home here is there are no significant interactions. Black kids are always at increased risk for asthma, regardless of where they live. And it's the same when you break it by ethnicity. Hispanic kids across all census tracts are always at increased risk for asthma, regardless of where they live. So why is that? So there's probably lots of reasons. One of the things that we wanted to look at was the role of racism. And in this analysis specifically, probably the most well-known codified example of structural racism, at least that we have, that we can look at in these types of studies, and that's redlining. So for those of you who haven't heard of redlining, this was a program from the Homeowners Loan Corporation. It was a New Deal initiative in the 1930s to provide mortgage refinancing to individuals in urban areas. And the way that they administered the program was they asked local real estate agents in 239 cities across the United States to go out and survey their neighborhoods and categorize every house into 4, or every neighborhood into 4 categories. And they did that with color. They drew maps literally in colors. And so they would draw a neighborhood in a city and code that as best, meaning those are the places that the loans should be made. They would do that in green. Then they had a category of still desirable in blue, definitely declining in yellow, and hazardous in red. So there's the term redlining. And basically the way they did this was the race, ethnicity, and immigration status of the residents of those neighborhoods. So those neighborhoods that were green or blue were the ones that got the mortgage refinancing. And if you look at the trajectories of those neighborhoods, the neighborhoods that were red, the hazardous and the definitely declining, were not eligible. There's gaps that exist to this day on health outcomes, on economic prosperity of those neighborhoods, to the location of where hazardous waste sites, to where interstates were built. All of those are associated with these red line neighborhoods to this day. So we wanted to look at here, because you know, if you think about it, why would it be that some realtor in 1930s outlines your neighborhood in red and that you are at an increased risk for asthma today? And what we wanted to do there was a causal mediation analysis. So the idea here is that the redlining changed the trajectory of that neighborhood such that now they have lower SES, a community level lower SES, and that then led towards the increased risk for asthma. So that sets up as a mediation analysis that I'm going to walk through here. And so we did this, we looked at, I'm going to show you the results for just one indicator of the neighborhood, percent of low-income households in that census tract. We did it for 5 different indicators and also some of the indices of deprivation that are out there, and the results are very similar. So I'm just going to show you the percent of low-income household results here. But the idea here, again, is that we can look at causal pathways by which redlining leads to the asthma through the neighborhood here. So the way a mediation analysis works, the first step is to show the left side of that triangle that there's an association between redlining and the percent of low-income households in a census tract. And you can see here that this is all in the CRU cohort again, in the homes of the CRU participants. The kids that grew up or were born in a census tract that was categorized as C or D indeed have significantly or are significantly more likely to have higher levels of percent low-income households in that census tract. And those that were not graded, so those are census tracts that actually weren't outlined in 1930 as part of the redlining program. So those are typically the suburbs of cities. So that those are the parts of the cities that didn't exist in the 1930s. Those kids are at decreased risk of living in a census tract that have higher levels of percent or higher levels of low income. And then the other side of the equation, we already saw this, but there's an association between the percent of low-income households in a census tract and the risk for developing asthma. In this analysis, the odds ratio was 1.16, so about a 16% increased risk per, we're doing z-scores here, so per standard deviation change in the percent of low-income households in that census tract and developing asthma by the time they were 12 years old. So we have both the elements set up for mediation analysis. We do have both sides of that triangle. And then when we run the causal mediation model, what we saw was actually no direct effect of redlining on asthma, but we saw a significant causal mediated effect, meaning the effect of redlining on asthma seems to be going through this pathway of, in this case, the variable percent low income household. So you can again see the C and the D, the kids that live in those neighborhoods are significantly more likely to develop asthma by the time they're 12 years old, higher in the D category, decreased risk for kids that live in the no grade category. So again, the suburbs for the most part. And importantly, you can actually estimate the proportion of that effect due to that mediation. And we estimate that about 30% of the effect in the C category is through that SES indicator variable of the neighborhood, and almost 80% is through that SES indicator for the D categorized neighborhoods. So there does appear to be a causal mediated effect by which redlining leads to asthma through that socioeconomic change in socioeconomic trajectory of that neighborhood. All right, so I'm going to completely switch gears now and spend just the last 10 minutes or so talking about some of the work we're doing with personal exposure to air pollution. So everything that I talked about so far is all based on these models of air pollution exposure, which have their strengths. We can do those with big electronic health record databases. If you have an address, you can estimate using these models the concentration of an air pollutant outside of their homes, right? But we know there's error in those models. We also know that what neighborhoods experience and what personal exposure you have is typically much higher than what you would measure if you were just to put an air sampler outside and monitor. And that's for a number of different reasons. One is just the activities of daily living, so the amount of time you spend in traffic. For kids, this is a study we did with an anti-idling program at Cincinnati Public Schools. You can see here the buses are lined up, they're idling. They're basically contaminating the cabin of the bus right behind them. So there's really, really high levels of ultrafine particles in those buses when we started. There's hot spots of exposure. There's indoor sources of exposure. Gas stoves is a big thing right now, and they do produce particles. And almost in all of our studies, children tend to have higher exposures than adults. They're outside more, they're lower, they're to the ground. They just tend to have higher levels of air pollution exposure. And so, new technology gives us the opportunity to look at what personal exposure kids receive. So this is just some pictures from a couple of studies. This is using a device you can see here that measures ultrafine particles. So if you're in the exposure world, this is a condensation particle counter. It uses water. It measures particle concentrations every second. And it has a GPS inside of it. And so we can put it on kids. This is just an example of two different kids, two different scenarios. The panel on the left is a child that lives in the downtown Cincinnati area right next to I-75. And basically you can see they have a relatively medium to high exposure from the time they left school to the time they got home. Contrast that to a different kid who is wearing our device, walks home in a suburb, and you see the high levels of exposure when they come up to intersections. So again, back to that stop and go traffic pattern where we think there's higher concentrations at an intersection. You can see it here in that figure. And the other advantage of personal sampling is you just get scenarios that you don't capture on models. So the other figures here are me wearing the device. I mowed the grass one day. You can see the particle exposure that you get while you're operating small machinery. So leaf blowers and lawnmowers and things like that. You're getting a pretty high exposure level. You don't capture on models. And that's all well and good. I mentioned, I'm not going to go into it, but I mentioned we did this study, anti-idling study at Cincinnati Public Schools. We published the results. It got a little bit of attention. I mean, it's not rocket science. Basically, if you turn off buses, the air quality improves outside of the schools. We showed it. And there was a local radio station in Cincinnati and talk radio, and they wanted me to come on, and my institution politely declined. And basically, they ripped me in the study because, what's it mean? What's it matter if a kid for 5 or 10 minutes walks from the bus inside their school? Forget that we actually showed the levels of air pollution inside the school actually increased because of the idling buses. But I didn't really have a good answer. I mean, the question is, it doesn't matter if you're standing outside when a bus comes by and you get a really, really high level of air pollution exposure. We can measure that with the personal monitors. But at the time, we didn't really know what that meant in terms of health effects. So that's where we've been thinking about and going with these panel studies that we've been doing more recently. The goal here is to actually look at much shorter term exposures because we can measure these pollutants in near real time down to the seconds. And so we can start to look at if we capture the health outcomes in repeated fashion over and over again. What does it mean for a very short exposure, for a short duration for your lung function? If you measure that lung function 7 or 8 times during the day, which we can do with all these different fancy tools that we have now. So this is a couple of studies that we have. These are some of the devices we use, but all of them are looking at short-term personal exposures to PM2.5 and ultrafine particles and respiratory health. We've now expanded this to measures of anxiety. So what does it mean over the course of a week and your fluctuations in air pollution exposure for your symptoms of anxiety? But we can do things like what's called ecological momentary assessment, which is basically repeated questionnaires on these are teenagers now in these studies, on their cell phones. If they answer, do they have respiratory symptoms? And we can trigger those questionnaires based upon when they got to school, when they first got up in the morning, those types of things. When we tie it into the cell phone, we can get repeated measures of the health outcomes and tie that in with our continuous measures of personal exposure. And so we've done this for like noise. We've looked at NO2, ozone exposures. We're interested in how it affects sleep. So we can get sleep data from putting a mat under their mattress, and it actually monitors their heart rate and respiratory rate, respiratory rate, and movement during the night and things like that. So we can start to tie in real-time exposure with more real-time health outcomes. And just a couple of the results. This is the analysis looking at 30-minute windows of exposure. So we have, 168 hours over the course of our sampling every second, a concentration of particles. So we can look at 30-minute windows here in this analysis. And this is looking at lung function data. So handheld spirometry, FEV1. And what you can see is that there's a decrease in FEV1 about an hour to an hour and a half after a higher level of ultrafine particle exposure in our study. And the effect, if we stratified this population, was 50-50 asthma, kids with asthma, teenagers with asthma, without asthma. The effect was greater in those with asthma. Same thing with symptoms. So the symptoms report was relatively rare, but we did see an increase about 30 minutes to an hour in ultrafine particle exposure with the risk of reporting a symptom. So this is shortness of breath or coughing. And saw that the effect again was higher in kids with asthma. So, this led me to think about what's our responsibility in terms of report back. So if you're doing studies of biomonitoring data, it's pretty, I think, universally accepted that you should be telling the study participants. You measure their PFAS exposure, you measure their phthalates, whatever it is in the blood. This is what we measure. This is the levels that we found in your sample. We, for the most part, don't do that in air pollution epidemiology, mostly because I think we're modeling exposure for most of these studies. There's error in the models, there's error in the time you spend inside, all those types of things. But you can see from those pictures, I hope that this is actually personal data that we're capturing on them. And I would argue, I think that it's even more informative than measuring, phthalate exposure in blood. Who knows, where you got that from in, 6 months ago when you took the blood sample and that. So I actually think, I would argue that the data we have when we're using personal monitors is more informative, and we ought to be thinking about giving that back to the study participants. And I think that it can inform action and improve education because of that. So we had a small R21 to actually do this. This was outside of my wheelhouse. We did focus groups, which I've never done before. It was really interesting. We actually brought the kids in from our panel studies, teenagers and their parents. And we asked them, how would you like to see this data? And we did a couple of iterations of this. A couple of things came out of this. Number one, they do not want paper copies. So most of the time when people give report back results, you get it in the mail, and it's like this big packet, and there's like a little test tube, and it says like, this is your level of phthalates. And the kids, the teenagers said, anyway, they're like that, we don't want that. We're not gonna open the mail. So they wanted electronic. We have geographic data here. So they want to see the maps, right? So that's what we developed. We ended up developing an HTML document that you can email everybody's personal results, which was a mix of information about, in this case, ultrafine particles. What is ultrafine particles? Background information. Here's your average level. And I can show after this, if you're interested, I can actually show you an example of the HTML document. You just scroll through, but it allows them to, I don't have a screenshot of it, but it basically gave them a tab for each day of their sampling, a map, which showed where they were when that sample was there. And then it showed a time series of the exposure. And if you hovered over either one of them, they would correspond to each other. So you could say, okay, I was at my house at this time, and this was my concentration. And for each day of their sampling, they could tab through and see that information. We did, I'm not sure here, I guess, we just published a short letter in EHP. We did a pre-post knowledge assessment on the kids and the caregivers in this study. So we gave everybody their result. We gave them the option of receiving their results after we developed the report back with the focus groups. They would do a pre-post knowledge assessment, and there are improvements mostly in the caregivers, their interest, their knowledge of where their kids were exposed to air pollution and what air pollution was. And so I think there's some promise maybe of using this as at least information that informs behaviors that could mitigate or at least reduce exposures if they know where they're exposed and what they're exposed to. I think the last thing I'm going to mention is the promise of low-cost air sensors. So these are two devices. If we were to go back and do the CAP study again, we spent, I don't know how much money doing the air sampling to get the PM 2.5, and then we estimated, we did that land use regression model on everybody. I mean, I would consider just giving everybody in a study now one of these purple air devices. They cost about $200, and you can put it outside of your home and actually measure PM 2.5 in real time. You can also upload the data, and as researchers, if they give you permission, we can download the data remotely. There's no home visits or anything like that. So there really is a lot of promise. They're quite accurate for some pollutants, not for every pollutant, but for some pollutants, and for PM 2.5, they're pretty good. We've used the purple air, and we also use something called the air beam for personal sampling. They both measure PM 2.5. They're both relatively accurate. They're deployable in networks, which gets you, if you're doing community-based research and neighborhoods are interested in their air quality, you know, the EPA regulatory sites are there for regulatory purposes. What's the general trend in the air quality in an urban area? Not necessarily my neighborhood that's impacted by this road or this factory. And so filling in those gaps, I think, is where a lot of the opportunity lies with the low-cost air sensors. People are developing networks, and we did the same thing. I won't go into a whole lot of detail, but this is a community in Cincinnati called Lower Price Hill. It's an environmental justice community, lots of health issues there. We worked with the community group that was housed there to establish a low-cost air monitoring network with the purple airs. And what we saw was that basically, if you were to use the EPA background regulatory sites in Cincinnati, you would miss about 5 days in the summer that we did the sampling where the AQI would have triggered an alert in that neighborhood that was not triggered if you use the EPA background site. So I think there's a lot of promise for this type of technology to be out there. There's a lot of interest in it. So we have a training grant. This is a shameless plug if anyone's interested. We have a training grant that's actually targeted towards academic and community partner teams that want to use low-cost air monitors in their research, in their projects. I think there's an unmet demand out there. There's community groups that don't quite know the technical limitations of the devices or how to deploy them. There's academic researchers who don't know how to do community research. And so the goal here is to actually provide that cross-training in both team science, community engaged research, but also some technical knowledge to use these low-cost networks. So we have an in-person training session. It's 3 days in the summertime for teams that apply to the program. We actually give them purple airs to get them started on their projects. And then we have follow-up training, webinars, and so on for a year or several years after if they want to continue to attend, to work through their projects and help them in their research. So just wrapping up, where are we going? Obviously, we have this cohort, the C4 cohort that we're still looking at now. I'm really interested in climate and climate change and the use of indoor air monitors to measure temperature and humidity inside of the homes. I think there's potential for interventions, even low-cost interventions with box fans, for example, that you can actually, you'll start to improve the air quality in homes and not just with HEPAs, which tend to be expensive and things like that. I'm interested in public health. How can we improve? If bad policy makes bad outcomes, maybe there's some good policies out there. And yeah, so I'll stop there. As a huge team that has worked with all of this, you know, at the University of Cincinnati, Children's Hospital, the CREW group has been fantastic to be a part of, and my colleagues at Harvard too that helped with the GIS stuff. So happy to answer any questions. Thank you. Yeah.
Your work looking at internalizing disorder,
yeah, and they're different for different outcomes. So yeah, we typically look at so in that chronic exposure, the CAPS one, there weren't differences, but we've seen them. Actually, we're looking at fluoride exposures in some of these, and we see sex differences with fluoride exposure. So yeah, there's definitely some sex differences depending on the outcome and the exposure that we're looking at. Yeah. So wonderful. Yeah, thanks.
Yeah, thanks. That was great. So when you were giving them the option of getting data back on air pollution, personal exposure, that's interesting because some other studies I know, all of us, they're giving data back. And you give them a choice. What percent say yes?
So full disclosure, it took us a while to get that grant and to go back to them. So it was about 2 years since they actually participated in the study. I have to go back and look, but I think it was about half of the parents wanted to get it back, and it was less than the kids, 30 to 40%. Yeah. But lots of those were not, no, we don't want to get it back. They just lost it. I mean, they're teenagers. They've gone to college and stuff like that. So I think that if we were to do it more, I mean, we would do that immediately after the study now. I think there'd be more interest having just participated in getting your results back. most of them like to see, the ones that did get it, thought, I mean, especially the focus groups are like, they want to see the maps. They're like, oh yeah, that's where I went to, that's my school. That's what my, the concentrations of air pollution are when I was at school, things like that. So they were interested in that, the technology component especially. Yeah. Yeah. Sorry, I was saying that the report back study took a little while. So there was a time gap between when they participated in the actual panel study and when they were offered the opportunity to get their results back. So I think that was a lot of the reason, but the desire for getting results was about 50% in the parents, the caregivers, and less so in the kids. But many of those were non-respondents that we had lost. So I think there's more interest. I think there would be more interest if we offered them the results immediately after they participated they participated in the study. Yeah.
There's a question online from Kennedy. First of all, she said it's a nice presentation. I'm wondering if the association of air pollution and child respiratory infections been analyzed. For example, hospitalizations.
Yeah, it has. We haven't done it. I've seen studies like that. COVID is a good example. There's been papers published with higher areas with higher levels of air pollution, higher COVID rates. So yeah, I think there's mechanisms out there that relate increased risk of infection. Of course, it's interesting too because areas with higher population density and crowded housing also have higher rates too. So there's some confounding there too. But yeah, the answer is yes. We haven't looked at it, but it would be interesting to take a look at. I don't know.
It's not a take, but I would assume that there's a relationship between pollution levels and temperature, humidity, barometric pressure, and you integrate those into. I mean, I was struck by that one slide. I don't know what happened on June 10th.
Yeah.
That type of
There-
thing. Do those things correlate?
They do, and it's interesting depending on the pollutant and where you are too. So for example, ozone is the classic example. You don't get ozone in the wintertime because you need high temperatures and sunlight and things like that. Secondary formation. Particulate matter is interesting. There's definitely cyclical natures. It changes depending on where you are. The sources of it change. So if you're doing a lot of burning wood, so in Montana, they have real high levels of PM 2.5 in the wintertime because they're burning wood versus, someplace like I imagine Detroit, but in Cincinnati, when the coal-fired power plants are the primary sources in the summertime because people are running their air conditioning. So you do get those relationships. And then there's also atmospheric transformations that play a role in that also. So there's not just one answer other than to say, yes, there is a relationship between meteorology and air pollution exposure. Typically, we're just looking at the estimated concentrations. We don't consider that in our epi analyses, but we do include things like season of birth, for example, more for, infectious disease, time of year and things like that. But yeah, so. You were first. Yeah. So I mean, that's kind of why the case crossover is so powerful because it's all within, a defined time period that you typically wouldn't have the changes in seasonality for. So I think that's evidence that there are certainly seasonality to the depressive symptoms. In the CAPS cohort where we're looking at, just one assessment of depression and anxiety, these assessments are meant to be validated across multiple time periods. So they're asking over the past 12 months, over the past, like long time periods of things to try to get around that. But yeah. Mm-hmm. Gray. So we saw decreased gray matter, and I don't have this. We saw decreased gray matter at 12 so the way we set up those MRIs is the kids that had the highest estimated exposures through the first year of their life and the lowest. So we had like a high and a low group, and the high group had less gray matter. We also saw on the spectroscopy data, oh my gosh, I'm drawing a blank on the, I'll get you the paper. There was a specific metabolite that was elevated that was also, we did a mediation analysis. It was associated with the anxiety outcomes. And I'm drawing a blank on it now. What was it? Yeah, it's myoinositol. Yes, myoinositol was elevated in that analysis too. So those are the primary ones that we've seen so far. We didn't see anything on the functional MRIs, at least with the air pollution exposures so far. Not in the gray matter difference. We didn't see that, no. So, but we'll see. I mean, we'll have, you know, MRIs at 12 and now in the early 20s. So we'll see if there's any changes over time. Yeah.
For exposure classification, have you tried to use a lag to go back into gestation?
We have, and the CAP study is not so yes is the answer. The way we did the recruitment was based on birth records. So the kids were actually 2 to 6 months old by the time we got to them. So we don't have, and we never asked them for a retrospective, like where'd you live during pregnancy? So we've done that assuming that they haven't moved since their birth record address. We haven't seen a whole lot of outcomes with the respiratory. Some suggestions with some of the neuro outcomes, but not a whole lot at this point. So we've tried, but I think there's a lot of exposure misclassification. We published a paper in the CAPS cohort. By the time they were 3 years old, about 40% of them have moved. So there's a lot of mobility in that population. So others have done similar work though. We just haven't seen that. Yeah. Yeah.
So obviously I'm a terrible human being, but I was an even worse child. I'm wondering if you comment on getting kids to be compliant with taking their personal monitors and wearing them in a consistent way.
I would say, depending on our study, so the ultrafine particle device that I showed is not an easy device to use. I would say that our sample probably isn't generalizable to every population. We recruited through Children's Hospital, emails to the employees. And so we have a relatively high SES and science interested group of kids in that study. The other devices though are much easier to use, much quieter. And I think more kids would be willing to use them, but it's just my supposition. We got pretty good compliance because I think of the group of kids that we had and the incentives that we give them. So, but we can measure, I mean, the nice thing about having GPS and we use some other devices that have accelerometers on them and stuff, you can measure how well they wear them. If it just sits there all day long, you know that too. So. Yeah.
We want to thank you so much.
Yeah, great to be here.